Hi, my name is Brianna Vargas Koya, and this is my Advanced Concepts Presentation 4 for Biochemistry 1. Thank you. For this presentation, I wanted to give an overall presentation prompt, discussion, an overview of what I will be discussing. So for this presentation, I will discuss every step that is involved in generating acetyl-CoA from pyruvate. Ultimately, in order to go from glycolysis to the citric acid cycle, we first must harvest pyruvates into acetyl-CoAs. This process involves many small steps which result in the main entrance in the citric acid cycle. This information, an overview of the pyruvate oxidation includes, at the end of glycolysis we have two pyruvate molecules that still contain lots of extractable energy. Therefore, the pyruvate oxidation is the next step in capturing the remaining energy in the form of ATP, although no ATP is made directly during pyruvate oxidation. Overall, pyruvate oxidation converts pyruvate, a 3-carbon molecule, into acetyl-CoA, as mentioned before, a 2-carbon molecule attached to coenzyme A producing an NADH and releasing 1 carbon dioxide molecule in this process. Acetyl-CoA acts as fuel for the citric acid cycle in the next stage of cellular respiration. Continuing on with the pyruvate oxidation steps. Pyruvate is produced by glycolysis, as we mentioned, in the cytoplasm. But pyruvate oxidation takes place in the mitochondrial matrix in eukaryotes. So before the chemical reactions can begin, pyruvate must enter the mitochondrion, crossing its inner membrane and arriving at the matrix. As you can see in this diagram, there's various steps, step 1, 2, and 3, regarding this entire process. Step 1 involves a carboxyl group, which is snipped off of pyruvate and released as a molecule of carbon dioxide. With that, it leaves behind a 2-carbon molecule. Step 2 involves this 2-carbon molecule from step 1 being oxidized, and the electrons lost in the oxidation are picked up by NAD+ to form NADH. Now continuing on with step 3, the oxidized 2-carbon molecule, as we mentioned before, which is an acetyl group, highlighted in green, is attached to coenzyme A, as we can see above in this diagram. CoA, an organic molecule derived from vitamin B5 to form acetyl-CoA. Acetyl-CoA is sometimes called a carrier molecule, as its job here is to carry the acetyl group to the citric acid cycle. The steps above are carried out by a large enzyme complex called the pyruvate dehydrogenase complex, PDC, which consists of 3 interconnected enzymes and includes over 60 subunits. At a couple of stages, the reaction intermediates actually to form covalent bonds to the enzyme complex. This becomes one of the cofactors. The pyruvate dehydrogenase complex is an important target for regulation as it controls the amount of acetyl-CoA fed into the citric acid cycle. Ultimately, the pyruvate oxidation is important for cellular respiration in order to improve our body, as well as circulation and other components of the system, including homeostasis throughout our body. Ultimately, a summarization of the pyruvate oxidation includes the following: 2 molecules of pyruvate that are converted into 2 molecules of acetyl-CoA. 2 carbons are released as carbon dioxide out of the 6 originally presented in glucose. 2 NADH are generated from NAD+. Overall, acetyl-CoA serves as fuel for the citric acid cycle, as I mentioned before. This leads on to the next stage of cellular respiration. The addition of CoA, acetyl-CoA, helps activate the acetyl group, preparing it to undergo the necessary reactions to enter the citric acid cycle. Overall, in the citric acid cycle, the acetyl group from acetyl-CoA is attached to a 4-carbon oxaloacetate molecule to form a 6-carbon citrate molecule. Through a series of steps, citrate is oxidized, releasing 2 carbon dioxide molecules for each acetyl group fed into the cycle. The purpose of this cycle is because it is at the center of cellular metabolism, playing a starring role in both the process of energy production and biosynthesis, as I mentioned before. It finishes the sugar-breaking job started in glycolysis and fuels the production of ATP in the process. I thank you for your time, and I hope you enjoyed my presentation for this week.